Professor Tuzinski, he did his postdoctoral fellowship at the University of Calgary Chemistry Department in 1983. He served as an assistant professor at the Department of Physics of Memorial University of Newfoundland from 1983 to 1988, and at the University of Alberta Physics Department from 1988 to 1990. As an associate professor from 1990 to 1993 and a full professor since 1993. He joined the Division of Experimental Oncology within the Cross Cancer Institute as an alert chair in 2005. He is on the editorial board of the Journal of Biological Physics, Journal of Biophysics and Structural Biology, JBSP, Quantum Biosystems Research Letters in Physics, Water, a multidisciplinary research journal, and Interdisciplinary Sciences Computational Life Sciences. He is an associate editor of the Frontiers Collection, Springer Verlag, Heidelberg. Professor Tuzinski delivered the inaugural talk at the opening workshop of DI Center for Consciousness Studies in October 2011. Professor Tuzinski.
Thank you very much. It's great to be back here after a year and a half, and thank you very much for the invitation. So today I'll be talking about two things. And first of all, I'll give a short perspective on what's happened in the last 15 years in regard to tubular and microtubules, which, as we heard, are playing a very central role in the consciousness studies these days. And in the second part, I hopefully get to it, I'll try to weave it into a longer perspective on the quantum physics leading to quantum biology leading to quantum consciousness. This is where I work, the Cross Cancer Institute, and this is exactly how it looks right now. So that's another reason I'm very glad to be here. The temperature yesterday was minus 15. So first of all, I'd like to thank my collaborators. This is the great pleasure of working with some great minds, and including Stuart Hameroff over the last 20 years or so, Travis, who is here, my former student, now an independent researcher, Avner Priel in Israel, Nancy Wolf at UCLA, Lloyd Demetrius at Harvard, Horatio Cantiello at Harvard, now Buenos Aires, Rudy Tanzi at Harvard, and Deepak Chopra, who's, I hear, coming tomorrow. You can find a lot of information about some of these ideas in the two books that I co-edited or co-authored, especially the one on the left, Nano-Neuroscience, which deals with some of the specific issues of microtubules in neuronal diseases, neurodegeneration, and other neurological disorders. So very briefly, microtubules are very important, all of eukaryotic cells, but they play different roles in different cells. So in a way, they are a multifunctional building block of life. In dividing cells, of course, they provide mitotic spindles and segregate chromosomes. I will not be talking about it today because it's a separate topic and requires an absolutely independent train of thought. And that leads, of course, to cancer and all these issues that I do on a daily basis, designing drugs for cancer. But what's more important, perhaps, in this context of the conference here is the role of microtubules in neurons. And they, of course, provide the mechanical strength to all cells and provide routes for transport, but they probably do much more. And with scratching the surface is about the surface regarding what they actually do in neurons. There is ample empirical evidence for the role of microtubules in almost all neurological diseases. And we've recently published some work on Alzheimer's, which Travis will probably spend more time discussing. But in all of these diseases, there is a common feature, which is a deteriorating cytoskeleton. And microtubules are either directly or indirectly involved in the neurodegeneration. Therefore, bioengineered cytoskeletal protein products or pharmacological agents can be and should be developed in the future to ameliorate these conditions. And I think The tiniest step made was the paper that we published last year with Stewart and Deepak Chopra and Rudy Tanzi towards solving the Alzheimer riddle. Currently, we're working actually on Parkinson's, where a similar problem exists, and we believe it can be solved as well. So in Alzheimer's disease, of course, there are multifactorial aspects of this degeneration, but all of it has to do with cytoskeletal protein disruption, and what is very well known is that hyperphosphorylation of MAP-tau leads to tangles and the formation of neurofibrillary tangles. But that's, as we presented in our paper, that's a side effect. The central role is played by dyshomeostasis of zinc, which leads to the breakup of microtubules and the accumulation of beta amyloids, which makes plaques fueled by the zinc stealing from the neuron. Anyway, Travis will say more about this. This is just words, but we have actual computational models. There are, as I said, direct or indirect involvements of microtubules and other cytoskeletal proteins in other diseases such as Parkinson's, where alpha-synuclein and parkin are mutated or misfolded and form aggregations. But they also impair axonal transport, and in Huntington's in particular, Huntington is disrupted in regard to binding to microtubules. I recently watched a movie about, actually it was a CBC show about traumatic brain injury, and I was very pleased to see actual experimental evidence of traumatic brain injury leading to the breakup of microtubules at the level of subcellular damage. So again, the cytoskeleton is disrupted. The same is shown in other diseases listed here, including epilepsy, ALS, and multiple sclerosis. So back to the theme of my talk. What have we learned in the last 15 years that we can use today and in the future to make progress in neuroscience as well as progress in understanding how the brain works? The first, and actually I chose 15 years for a reason, because in 1997, a breakthrough occurred with the final achievement of crystallography of tubulin by the group of Downing at Berkeley. This was published in 1998, so you can argue it's still 15 years. And since then, several crystal structures have been published of tubulin, and we've been working on these structures, trying to understand how structure informs the form and how this, in turn, affects the function of tubulin microtubules. Actually, you can do better than that, because with current computational methodology, you can make predictions regarding tubulin structures. Tubulin is not just one protein, it's a family of proteins, in fact, expressed by different genes. There's close to 20 genes for tubulin in the human body, and each cell uses it differently, and different organisms utilize different tubulin sequences for specific functions. This is still an open issue. I think it will take maybe decades to disentangle the enigma of tubulin. In spite of great homology between species, with regard to tubulin, there are subtle differences which affect the function. And this we can do from the sequences. Knowing the sequences of amino acids, or in DNA, nucleic acids, encoding for tubulin, we can construct three-dimensional models of the protein and understand the interactions between tubulin as they make microtubules. We've done that. As well as to understand how different ligands or other proteins, and there's hundreds of proteins interacting with tubulin, are affected by these changes. So atomistic detail is possible. We currently work at the level of single atoms, knowing exactly where each atom resides in the structure of tubulin, how it dynamically interacts with others, and the environment. And from this, actually, we can even go deeper into the quantum level. Okay, this is just an example to give you some sort of mental image of how deep we can go with this. This is a colorful representation of the distribution of different tubulin isoforms. Each isoform is encoded by a different gene. The top panel shows you something like 17, I think, or close to 20 tissues, normal tissues. So if you look at, let's say, the brain, the first one on the left, and compare it to prostate, you have a vast difference in terms of which tubulins are used in these different organs. So it's not just tubulin, it's the spectrum of tubulins to choose from. Each tubulin isoform behaves somewhat differently because of the structural differences. The bottom panel shows you distribution of tubulin isoforms in cancers, different cancers, from cancer of the larynx to lung to kidney to colon to breast to ovarian and so on. So we also have to abandon the current paradigm that that one drug will solve one, we will find a silver bullet that will cure cancer with one drug. This is not possible, simply because different cancers have different phenotypes, even in regard to the tubulin involvement. So why is it important? It's important because now we can link single point mutations to structural and functional differences of tubulin. We have built atomistic models of tubulin and can use these models to design drugs for various diseases. And by the way, what I said about cancer can be translated also into neurological diseases. And currently, by the way, there is not a single drug, to my knowledge, that targets tubulin in neurological diseases. So there is a vast area opening up. Even a year ago, there was a review paper published on examining the current cancer drugs vis-a-vis neurological applications. We don't have anything there yet, but it's doable, it's possible. So we have already designed specific drugs and can bioengineer intracellular proteins as well. What's very interesting as well is that without an exception, all drugs that currently target tubulin originated as extracts from natural products. So plants and microorganisms, bacteria, produce toxins that we can use as blueprints for affecting human tubulin. So that's another area where we're actually working very actively in. At my university, University of Alberta, we have just completed a monumental project of sequencing 1,000 plants. We have the DNA of 1,000 plants, including the sequences of 1,000 tubulins. Just to give you a perspective, until now, if you look into the gene bank to look at the sequences of DNA from all species known to man, there's something like 300 tubulin sequences, maybe 320 on this order. We have added 1,000 to it, just from plants. And this is being sifted and analyzed. It will take years to understand it all. So why is it important? Right. Because we can not only design drugs for tissues, but we can make it patient-specific for cancer or other diseases. Because mutations will be specific to a group of patients or subpopulations. We can also design drugs that overcome resistance due to continuously evolving phenotype of the disease, especially cancer is mutating rapidly. And finally, using these plant sequences, we can mimic Mother Nature's solutions because these plants are immune to their own toxins. In other words, it's a blueprint for resistance to drugs, and cancer cells or other diseases will try to, by mutations, achieve this solution. So let's now focus on microtubules. So keeping in mind that tubulin is virtually completely understood, we can build models of supramolecular structures from these dimers of tubulin as building blocks. And this is a very difficult task to some degree because microtubules are dynamic. They have something called dynamic instability, which is constantly growing and shrinking polymer, and it's growing and shrinking asymmetrically. That is, the growth is linear, and the shrinkage is catastrophic, nonlinear. So it's also a stochastic event, an earthquake-like event in microtubules. But of course, in living cells, cells overcome this and harness it, and they have assisted proteins that assist in the stabilization of proteins when needed, and destabilization in other situations. So this just shows you a molecular model of a difference between a growing protofilament of tubulin, of a microtubule on the left and shrinking on the right. I want to emphasize one aspect here that I think in the context of consciousness, I don't want to hammer this point, no pun intended, but non-equilibrium, life is a non-equilibrium process. It depends on energy. And microtubules are also non-equilibrium polymer systems, which are crucial depending on energy supply. If you stop the energy supply, they will simply disassemble and disintegrate. So constant supply of energy is necessary for living systems to survive. It's also the same for microtubules to continue performing this interesting dance of growth and shrinkage. That's another view of the same depolarization process that can be stabilized by the supply of energy at the GTP cap. And that's a completely reconstructed microtubule, including C-termini, which play an important role in communicating with the environment. All the motor proteins and other proteins like MAPs attached to microtubules with C-termini, these little whiskers that decorate the surface of microtubules. And by the way, these whiskers, the C-termini, are a short sequence of amino acids that has a unique address. It's like your name and my name. They're unique. Every tubulin has a different C-termini sequence. So different species, different isoforms, etc. And they can be also altered by so-called post-translational modifications that are now known to perform a major function in disease development, including cancer and neurological diseases. So somebody mentioned before that microtubules exhibit two types of lattices. They differ in symmetry and differ in energy. Now let's switch gears a little bit and go into something that will be expanded upon by Travis in a few minutes. And this was actually a discovery that Stuart communicated to me, and we worked for about 2 years on trying to understand what it means. These researchers, I think at Johns Hopkins, if I remember correctly, no? East Coast somewhere. All right. So in 2008, at the end of 2008, they discovered that they can erase memories of mice by inhibiting calmodulin kinase 2 enzyme. And so we went from this point trying to understand what this can do, and basically discovered somewhat accidentally that there may be something called the memory code, namely microtubules can be phosphorylated by CAMK2 in various patterns, and these phosphorylation patterns serve the purpose of encoding memories in a similar fashion to bits in a computer, but these are trits or triple states. We published this last year in PLOS Computational Biology, and Travis will explain the model in great detail, but I just wanted to say that there is a connection to memory through this process, and we heard this before. I just want to emphasize again, this type of memory could be erasable, could also be encoded by other processes listed here, by C-termini states, which we know from simulations exist in, and also from experiments, exist in at least 2 conformational states, through electron hopping, because of the tunneling potential inside the dimer, through conformational changes in the dimer itself, and phosphorylation of the states, which I just mentioned. So if you combine this, you obtain at least 128 possible states per dimer, at least. And some of these states actually can be inherently quantum, such as electron and also exciton states, which I didn't count. But even with these 128 states per dimer, you end up with an enormous storage potential in the human brain, which amounts to something like 10 to the 20th bits per brain. And I've read recently, and probably you know about this, that there is ample evidence that our brain stores all memories and keeps them forever. There are not only autistic people that can recall events in great detail, including perfect quotes from books read 20 years before, but also under hypnosis, you can recall events in great detail and recall memories. So when people doubt this, I say, actually, this is not so far-fetched, because there is empirical evidence that our memories can be very detailed and can be enormous. And where can you store this? Well, this is one possibility, and I think we have a very good feel for how this can happen. Now, the question of how fast you can access this memory and how rapidly that can change, that's still an open issue. If you take a microsecond, which is a typical time scale for protein conformational changes, then you get something like 10 to the 26 flops. The most powerful computer today, the IBM's Blue Gene, has the operational speed of 10 to the 15 flops, which is 11 orders of magnitude, 100 billion times slower than this, than our human brain if we used it to full capacity. So that gives you a perspective. I may be overly optimistic, but I think I don't want to be overly pessimistic like some other. I will not name names. So why is it important? And again, try to be a little bit practical here. I believe that in maybe not in my lifetime, but in this century, we'll be able to perform memory augmentation if we understand these processes in detail. We'll be able to enhance memory. Maybe we'll be able to selectively erase memories, because some memories you don't want to keep. And finally, you may be able to, or we may be able to export memories into somebody else or some other devices, so you can share memories in a very intimate way. I always, just as a side note, I always find very difficult to convey to somebody else experiences I've had. For example, when I come back from India, I try to explain what it was like. It's virtually impossible to explain it, because the amount of impression that you get and the whole experience is so singular that you give up and you say, well, you just go to India and you'll find out. So another piece of work that Travis and Stewart and I performed last year and published also in PLOS One is, and this is Stewart's domain, his lifelong quest to understand anesthesia. Travis performed blind docking of anesthetics to tubulin, and we found numerous set of binding sites for anesthetics. We've done some experiments as well on one particular anesthetic, halothane, which confirmed one of our predictions. And I heard here that some other researchers at Dalebag are working on an anesthetic. That's a great field to enter into vis-a-vis microtubules. So Stewart had a hunch, and I think we are confirming computationally that anesthetics actually play a role by binding to microtubules. And what will they do? So now we have to speculate a little bit, and I think they affect quantum states that exist in microtubules. This is Stewart's original idea. We're now trying to put it in practice. This is unpublished work. We've also done blind docking of psychoactive drugs. As some of you may know, our brain functions differently under the action of these compounds. And I'll just say that I'm very familiar to caffeine and chocolate, and I like both. I don't know any other things, but we can do experiments on those. And we found that all of these compounds also bind to microtubules in different locations from anesthetics, actually vastly different, and actually they bind much more strongly. So they may have a stronger effect. And again, what does it mean? These are the drug binding sites known to us, and there's one more which is missed here, but I couldn't find a picture. There's 4 known binding sites, one for vinblastine, one for paclitaxel, one for colchicine. And these psychoactive drugs also tend to bind in these locations. So to put it together, we have performed computational predictions and have some partial experimental confirmation, but we're still looking for collaborators who could do more experiments on psychoactive and anesthetic molecules vis-a-vis microtubule binding. And they also suggest involvement, and in one case, enhancement of cognitive functions or maybe acceleration of cognitive functions, namely psychoactive. In other, complete removal of cognitive functions, anesthetics. So Stewart proposed a hypothesis which involves the quantum states of tubulin are being involved. In one case, enhanced, in other words, transitions between quantum states could be made more rapid or stronger, so psychoactive drugs. In the case of anesthetics, they can be quenched. So quantum transitions are either slowed down or completely stopped, temporarily, luckily for us. So anesthetics quench quantum hopping, psychoactive drugs enhance quantum transitions. This is actually in line with some of our earlier work going back almost a decade. Now, I'll get back to this in a second, but I want to say that what does mean quantum transitions? And we can see at least two possibilities. One is exciton hopping, that means dipole transitions, quantum dipole transitions between some groups in tubulin. The other is electron tunneling. So the difference being that dipole transitions don't involve any mass transfer, only state transfer. Electron tunneling means actually charge and mass transfer, mass movement along the microtubule. Both of these can be tested experimentally and should be. And I'll tell you why it can be done, but you have to wait another 5 minutes. So I think in 2003 we published, Stewart and I, we published an analysis of possible quantum exciton pathways in microtubules, which would involve amino acids which have benzene rings that are known to be excitable by fluorescence. And they also are involved in quantum hopping between them. And they're actually all within something like 20 angstroms, which is acceptable distance for quantum hopping. We published this in 2003, I think, in Biosystems, and both of us were subsequently ridiculed by various colleagues at presentations. I think now it's coming back and actually is making a stir, and the whole field of quantum biology is being built along similar lines in a different system, and you'll see it in a second. So these are tryptophan pathways in tubulin on the surface of microtubule. So just several. You could imagine numerous types of possibilities for the movement of these quantum oscillations, each of which could correspond to a different type of excitation, maybe a different mental state, or something like this. There is a vast literature, and this is only a sampling, demonstrating the role of tryptophan amino acids in both excitonic or dipolar and electronic, namely charge transfer at the quantum level, and all of this involves migration of energy in proteins and in enzymes. So that is completely accepted. It happens in various cases listed here. Travis worked on an early model of electron transfer before we actually focused on exciton transfer, both of which are still real possibilities, which started from the electrostatic surface cross-section across the dipole of dimer of tubulin, and we found the possibility of a double potential well with a barrier that can be overcome by quantum tunneling of these electrons. And that's a cartoon, the way physicists see this situation from the point of view of an electron sitting either on the left or on the right of the barrier and being able to tunnel over it. Now, the question is, will it tunnel or will it not? It all depends on roughly two things. Firstly, what temperature is it at? And secondly, what is the height of the barrier? And that determines the probability of hopping. And so Travis did quantum Monte Carlo simulations on a microtubule lattice and examined a spectrum of parameters that were physically realistic. We didn't have all the numbers. This was based on Coulomb interactions and simultaneous updating rules with the probability of hopping given by this formula here, Boltzmann, basically. Formula with quantum states separated by delta E. And there were 4 possible states that could emerge from this pattern: homogeneous, static, or oscillating, chaotic, and stable moving structures. This is the last one is what we wanted to prove, because actually Stewart wrote a paper in 1982, if I remember correctly, on something similar at the classical levels, cellular automata. We wanted to see if his hunch was correct again at the quantum level. And so this is if you can see it, these are the results at room temperatures, and we assumed different values of dielectric constant. At that time, we didn't know what it was. Later on, it was measured actually at 8.41 for microtubules. And you can see the pattern for B, for A, and for D, for C as well, in various slices of this phase space. So there exist possibilities depending on the potential well depth, and something on the order of 100 to, I don't quite see the numbers here, 120 millivolts, which is a realistic assumption, is potential leading to quantum Monte Carlo transitions propagating to the microtubule. So the answer is yes, it's possible under fairly restrictive conditions, but still possible. Okay, this is now, I'll try to move on to the second part of the talk. And that's the concept I want to discuss is quantum consciousness, quantum physics, 2 extremes. In the middle, we have quantum biology, that is the bridge between the two areas of endeavor. And quantum biology right now is becoming a very hot topic. And I think this is an important topic also in the context of this conference. And there was a question earlier this morning about decoherence and things of this nature, which is very common to ask. Almost everybody who thinks about this will ask the same question. But I want to remind the audience that quantum physics is not just electrons and photons and neutrons. Quantum physics actually has macroscopic manifestations. We've been there for the past 100 years or more. And in quantum solid-state theory, we're not surprised that things can be operating at a quantum level even at room temperature. This is actually quite common. I'll give you one example that is ancient: magnetism. Magnetic substances cannot exist in classical physics because of spin. Without spin, there is no magnetism. So this is a macroscopic, room temperature, even higher phenomenon, which is absolutely quantum at the core. Another example is crystal vibrations. Crystal oscillations cannot be explained without resorting to quantum statistics. This was first attempted by Einstein, and he actually failed at this. He got the wrong answer. And Paul Debye solved the problem in 1930 and got the Nobel Prize. So at room temperature, crystals vibrate, and if you look at the specific heat of solids at room temperature, you will see signatures of quantum behavior. Lasers, I mean, these days we use lasers every day, even here. I don't know how to use it, but so lasers overcome the problem of decoherence at room temperature by being pumped by energy. So once again, I told you earlier that energy is all important in life, and the combination of macroscopic nonlinear interactions and formation of condensates plus energy pumping can easily overcome many of these objections. But I think most of the people who talk about quantum biology think about quantum weirdness, these crazy phenomena that only exist in the quantum world, such as entanglement, spooky action at a distance, etc. And even that is currently being seen as possible at the level of room temperature, physiological temperature, biological systems. And I think currently biology is exactly at the same point that physics was at the turn of 19th and 20th century, 100 years ago, 110 years ago. There are several unexplained phenomena in biology that are difficult to include in the fundamentals of biology, but they could be explained with quantum principles, but people resisted, of course. In physics, Max Planck, who was actually internally opposed to quantum mechanics, he couldn't explain blackbody radiation without introducing energy quantization. Blackbody radiation is very similar in a way to one aspect I will hopefully be able to discuss, namely metabolic rates of living systems. And the other phenomenon that actually Einstein solved and got a Nobel Prize for was the photoelectric effect. The photoelectric effect involves ejection of electrons from valence states in metals to the conduction band by absorption of photon energy. So this is one of those experiments. One particle, electromagnetic quantum of light, strikes the surface and ejects another one. This is completely analogous to what now is causing a stir in biology, namely the photosynthetic phenomenon. I think I have a slide in a few minutes about this. So photosynthesis is very similar in a way because light causes excitations to arise in the chlorophyll. And these excitations are funneled and gathered at reactive centers. So I just listed here some terms that are associated with quantum biology, which basically is a field that links quantum physics with life sciences. And in some directions, which I have no time to discuss, but you can look up, vision. Vision is our sense of vision is inherently quantum because a single photon of light or two photons can be detected by the retina. This can then be amplified through a process of conformational changes and current amplification and basically registered in the brain. So I think, to me, this is a very good example of how we should think about how biology can integrate all of these crazy ideas. They're not that crazy when you think about going from basic fundamental quantum event through amplification to a classical observation or registering of that event in the brain. Okay? Quantum olfaction, the sense of smell, the same, but I have no time to discuss it. You look up Luca Turin, and his work is amazing, as I said. So quantum metabolism is very interesting because it really mimics what happened in physics in the 1930s, specific heat of solids. I will skip this in the interest of time, as I have only 5 minutes, right? Just bringing this to the attention of the audience, that cells have been demonstrated to react to photons through microtubules in the centrioles. So centrioles are composed of microtubules, 9 triplets of microtubules with a doublet in the middle. And that's a very good example of how one process, which is capture of a photon, leads to a macroscopic movement of a living system. I'll leave this aside. Just a historical note that Herbert Froelich, some 30 years ago, launched a meta-theory, basically, which deals with quantum coherence in living systems. And this is, again, very similar to laser action because it requires energy pumping. And I keep sounding like a broken record, energy, because that's what life is all about. So life needs energy, and there are only 2 sources of energy. Light for plants, nutrients for animals, and the outcome is quanta of biochemical energy, ATP molecules. This is the currency of life in terms of energetical content. So bioenergetics is a quantum phenomenon, both for plants and for animals. For plants, is now agreed on because of the photosynthesis theory that was now published in Science and Nature in these high-profile publications. The neglected player in all of this is the mitochondrion, the energy-producing organelles in animal cells. I'll skip this. So this is an arrangement of these in this complex of the ring-like molecules that capture photons and create excitons and share it and produce coherence. We now know on the order of 1 nanosecond or more. So quantum coherence exists in plants. Why can it not exist in animals? So I'll close now with this foray into a little bit of math and a little bit of physiology. Quantum metabolism. This is a term coined by my collaborator Lloyd Demetrius at Harvard and Max Planck. The formula at the top is called the Kleiber law. It's been known for almost a century. A French scientist, Kleiber, analyzed metabolic rates of numerous organisms across all species. And he came up with the scaling law, which is written here. Basically, it's an allometric scaling law. Metabolic rates do not scale with size. If you double the size of an organism, it does not consume twice as much energy. It consumes less than that. Typically, 2 to the power 3/4. Typically, but not always. As you can see at the bottom, the metabolic rates for unicellular organisms scale with 3/4, for plants between 2/3 and 1, for animals between 2/3 and 3/4. Very peculiar. And there were many papers published in the last decade or two on trying to explain it. They all failed because they assume vasculature is necessary, so it's limited by transport. It's not, because bacteria don't have vasculature. Single cells don't have vasculature. Just like Stewart's argument about paramecium, that's the same argument here. You can explain it by assuming that metabolic enzymes form a network. They're embedded in the structure, form a network, and undergo electrochemical oscillations, which is all very well known. Mitchell theory, Nobel Prize, 1983. And there are only 2 assumptions necessary to explain all of this with one stroke. That is, no quantum physics. Go back to solid-state theory and read Debye's papers. And use quantization principle for metabolic enzymes, which we know makes sense because they do have a quantum of metabolic energy, which is ATP. It's actually about, 60 or 80 kilocalories per mole. I'm almost done. 2 more slides. But that's the punchline, so I don't want to miss it. Debye explains specific heat of solids by assuming something very important that Einstein did not understand. If you have quantum oscillators in a network, they synchronize. They form synchronous oscillations and actually form waves of oscillation. And if you, instead of integrating over oscillators, integrate over all possible waves that can occur, you end up with the correct scaling law for the specific heat of solids. That was good enough for a Nobel Prize then. It should be good enough for a Nobel Prize now. You do the same thing for metabolic enzymes. You do the same integration, assume quantization of metabolic energy transduction, and assume different dimensionalities because these metabolic enzymes can be distributed in 2 dimensions in a chlorophyll or 3-dimensional space in a tissue. You end up with this law, which is the basal metabolic rate P on the left scales with the mass to the power d over d plus 1. D is dimensionality. When you put d equals 1, you get 1/2. If you put 2, it's 2/3. If you put 3, it's 3/4. If you put infinity, and I will leave it as a puzzle for all of you to think about, you get 1. So you spend absolutely all possibilities known to us in the animal kingdom and in plants. And I'll finish with one thing. If you analyze the result, this is published work. You look up Demetrius and Tuzinski. If you analyze the results and find out what this constant that is required to solve the puzzle of allometric scaling laws in physiology is, it's like Planck constant. Planck constant was first obtained empirically from black body radiation, and it turned out to be a fundamental constant. We did the same thing for living systems, and the value is 11 orders of magnitude, 100 billion times bigger than the Planck. Why can this be? our answer is that a metabolic enzyme is not a single atom. It actually serves as a sphere of influence which comprises that many atoms in a cell. And I want to stop at this point because thank you very much for your attention.